Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, let me introduce my team. We are from group 1. There are Ahmad R. Akumali, Melisa Rahman, Widyastuti Hahabi, Desi Nuraini Lakutu, Dini Minarti R. Sulemante, and Melanie Frisilia Makidong. In this experiment, with the title is Kualitatif Analysis of Secondary Metabolites in Plants in Meranti. And the objectives is to determine the content of secondary metabolites in plant in Meranti. And the literature review is the phytochemical test is an examination of the class of chemical compounds contained in plant Symplesia. This then can be used to prove the presence or absence of the certain chemical compounds in plants to be associated with their biological activity so that they can assist phytopharmacology steps. And the experimental that used in this, the tools used in this experiment, they are mortar and vessels, fabric tin, pipettes, scissors, drip plate, test tube cells, test tube, aluminum foil, test tube holder, and the last one is Bunsen burner. And the experimental material is water, hydrochloric acid, concentrated of sulfuric acid, magnesium, chloroform, iron trichloride, Meyer's reagent, Wagner's reagent, Dragendorf, sodium hydroxide, methanol, acetic acid anhydride, grape leaves, discoriated hispida leaves, alpino Newton leaves, and fern leaves. And the sample used in this experiment is Vitis vinifera, Piper reticulatum, Alpinia notans, and the last one is Polypodiopita. And the process of extracting sample in this experiment, the sample was firstly measured or caressed, which serves to damage the cells walls of leaves. Then the sample was diluted with aided aqueous and filtered and squeezed to extract the extract. Then it was carried out through several qualitative tests, namely the flavonoid, alkaloid, steroid and terpenoid, saponin and tannin test. In the flavonoid test, after adding magnesium hydrochloric acid, concentrated of sulfuric acid, and sodium hydroxide, it turns orange and there is a foam. On the addition of the magnesium hydrochloric acid, this indicates that the plant 1 is positive for flavonoids. The purpose of adding magnesium and hydrochloric acid is to reduce the benzopyrene core contained in the flavonoid structure. In addition, the addition of concentrated hydrochloric acid is used to hydrolyze flavonoids into their glycans, namely by hydrolyzing or glycosyl. Glycosyl will be replaced by H+ from acid because of its electrophilic nature. Reduction with concentrated magnesium and hydrochloric acid can be produced complex compounds that are red or orange in color in flavonols, flavonones, and xanthanones. In the alkaloid test using Dragendorf, Wagner, Hager, and Meyer reagents and adhered hydrochloric acid, the results showed that plant 1 was positive for alkaloid compounds. For Dragendorf's reagent, it turns orange and a precipitate is formed. Wagner becomes dark green, Hager is clear blue, and there is a precipitate, and Meyer becomes yellow to form a precipitate. With the addition of hydrochloric acid will be increase the solubility of alkaloids because the alkaloid compounds will react with hydrochloric acid and will be form soluble salts. In addition, the addition of adding hydrochloric acid is because the alkaloids are alkaline, so they are usually extracted with acid containing solvent, while the heating function is to form a stable alkaloid salt. In the steroid and terpenoid test, 0.5 ml of chloroform, ammonia and acetic acid were used. Lieberman-Borsod reagent and the concentrated of sulfuric acid were 10 drops each. The result obtained were that the plant 1 was positive for steroid because a green precipitate and ring were formed. The addition of chloroform aims to dissolve the steroid compound in the sample. Steroid compounds will be dehydrated by the addition of concentrated sulfuric acid and form ions that give a number of color reactions. The color change that occurs is due to an oxidation reaction in the steroid group through the formation of a conjugated double bond. In the saponin test, 10 ml of hot water was added to the sample extract to produce a yellow foam. This indicated that the plant 1 is positive for saponins. Foam formation can occur because saponins have two opposite properties in their structure, namely polar glycosides and nonpolar steroid rings. So when shaken with water, only glycoside will be to water, while the steroids will repel water. This is the principle of foam formation. In the tannin test, the sample extract was aided with iron trichloride, so that it changed from a green color to a blue-black color. This indicates that the plant 1 was positive for pyrogallol tannins. Tannins are phenolic compounds that tend to be soluble in water and polar solvents. The purpose of adding iron trichloride is to determine whether grape leaves contain a phenol group. The presence of phenol group is indicated by blackish green and blackish blue after being added with the iron trichloride. The flavonoid group in the flavonoid test that were several tests, namely flavonoid testing with concentrated of hydrochloric acid and magnesium powder, the sulfuric acid solution, and sodium hydroxide solution. Based on the observation made, positive results were obtained from tests using sulfuric acid solution, which produced clear green, while the result of the test using concentration of hydrochloric acid and magnesium powder samples turned yellow and frothy. The alkaloid group in this experiment, the test was carried out with several reagents, namely Meyer's reagent, Wagner's reagent, Drag-and-Drop reagent, and Hager's reagent. The result obtained from testing with this reagent were positive. This is because in the Weyer's test, a clear green solution forms a precipitate. In the Wagner test, a pale green solution from a precipitate. In the Drag-and-Drop test, a bright green solution from a precipitate. And this is shown in the following figure, in the right figure that you can see. The saponin group, based on the result of experiment carried out by samples of the thick extract of pipereticulatum leaves, was negative and did not contain saponins, which was indicated by the absence of foam and bubbles. The steroid or terpenoid group, based on the results of experiment carried out by samples of thick extract of pipereticulatum by the leaf extract, was positive for terpenoids, which was indicated by the sample forming a green ring. The tannin group, based on the result of this experiment carried out by sample of the thick extract of the pepper reticulatum leaves, was positive for tannin, which was indicated by the change in color to a blue-black color. In the flavonoid test, after adding magnesium hydrochloric acid, concentrated of sulfuric acid, and the sodium hydroxide, it turns orange and there is a foam on addition of the magnesium of hydrochloric acid. This is indicate that the alpineanotant is positive for flavonoids. The purpose of adding magnesium and hydrochloric acid is to reduce the benzopyrene core contained in the flavonoid structure. In the steroid and terpenoid test, 0.5 ml of chloroform, ammonia, and acetic acid were used. Lieberman-Borsted reagent and the concentration of sulfuric acid were 10 drops each. The result obtained were that the alpineanotant was positive for steroid because a green precipitate and ring were formed. In the flavonoid test, with concentrated of hydrochloric acid and magnesium powder, sulfuric acid solution, and sodium hydroxide solution, based on the observation that positive results were obtained from tests using the sulfuric acid solution, which produced a brownish yellow color, and the sodium hydroxide solution, which produced clear green, while the result of the test using concentrated of the hydrochloric acid and magnesium powder sample turns yellow and fruity. In the steroid and terpenoid test, based on the result of this experiment carried out by sample of thick extract of Polypodiophyta leaves extract, was positive for terpenoids, which was indicated by the sample forming a green ring. So the conclusion of this experiment that the secondary metabolites are energy and food stores in plants and can be used when needed. Based on the result of this experiment, it was found for the sample ABCD, consisting of Vitivitifera, the grape leaves, Pepper reticulatum, alpineanotant, and firm leaves or the Polypodiophyta. With each test, it can be concluded that for the flavonoids, changes color positive containing flavonoids. For the alkaloids test, there is a color change and precipitate is formed, meaning that it contains positive alkaloids. On the other hand, if there is only a color change but no precipitate is formed, it means that it does not contain alkaloids. For the steroid and terpenoid test, if the sample forms a precipitate and forms a red or green ring, it means it is a positive steroid and terpenoid. For the saponin test, if the sample produces foam and changes color, it means the sample contains saponins. And for the tannin test, if there is a color change, the sample is positive for the tannin.